So now in this fast, furious coverage of sexual reproduction, we need to get to the point of sexual reproduction, and that is genetic variation. Ultimately, this idea that I need to find another organism to reproduce is actually kind of ludicrous. When I'm an asexually reproducing organism, I'm not dependent on finding anything else. I can make more of me. That's how bacteria do it, right? That's very, very successful. But evolution has selected for this mechanism because it promotes variation. And with variation in a population, that population is more likely to survive based on natural selection. And as you study evolution more, you'll see the power of this genetic variation and a mechanism to maintain it, to literally get in there and mix up the genes in a population. That's what this is all about. And we're going to look at this more carefully now as we compare mitosis and meiosis, and then look at the specifics of meiosis and how we generate variation. So again, the whole point of mitosis is to make a copy. I want a cell that looks like me. I want another copy of it. So let's look at this chart. This is a great slide. I love this slide. Look at your parent cell. It's a diploid cell. It's 2N. It has 6 chromosomes. If I'm going down the left side, mitosis produces 2 cells that look exactly like that parent cell. Prophase, metaphase, anaphase, telophase. PMAC kicking in. Boom, there you go. Meiosis, on the other hand, is going to produce daughter cells that are half the chromosome number, 1N. And the chromosomes are mixed up. So we go through meiosis 1, get crossing over, we got metaphase 1, homologous pairs line up, not just the chromosomes, and then we have the ploidy. Boom, we're down to only 3 chromosomes, right? So very, very side-by-side comparison is very helpful. This chart can do the same thing. So this is a slide that you can come back to and study when you want to think about the differences between comparing and contrasting mitosis with meiosis. Now there are 3 events unique to meiosis, and again, like I said before, it's all kind of front-loaded. Most of this stuff is going to happen in all this stuff is going to happen in meiosis 1. And so those 3 events are synapses forming, so synapsis, and crossing over in prophase 1. Literally, chromosomes connect and exchange genetic information, but homologously. On the metaphase 1 plate, homologous chromosomes pair up instead of individual chromosomes, right, like we saw in mitosis. So we have now pairs lining. It's like instead of single file, we've got double file, right? And that double file means that those 2 lines can walk away from each other laterally, and that's going to have the number. Anaphase 1 is where this is going to happen, right? So those are our big 3 events. And what this is going to do is increase genetic variation among the offspring. And so we can kind of drill down to what we call independent assortment, review crossing over, and then the idea of random fertilization. Okay, so independent assortment is really important, and Mendel, which we're going to talk about later, kind of the father of modern genetics, Mendel was able to figure this stuff out even without knowing anything about DNA. But what we're going to see is that we can understand it on a molecular level, on a chromosomal level. So we're going to look at it on that level first. So the idea is that each pair of chromosomes, when they line up on the metaphase plate, it's random, whether they're on the left or right or left. Let's look at that. So again, you've got many, many different combinations when you independently assort. And actually that number is 2 to the 23rd. That's more than 8 million possible combinations of chromosomes lining up on the metaphase plate. Here is an example of just a 4 chromosome organism with 2 pairs of homologous chromosomes. So possibility 1 is you have both of dad's chromosomes on the left and mom's chromosomes on the right, or the first pair, dad's chromosome could be on the left and mom's chromosomes on the right, and the second pair, mom's chromosomes on the left and dad's chromosomes on the right. So what does this lead to? This is random. So for us, instead of seeing just 2 pairs, you'd have 23 pairs, and so that's where your 2 to the 23rd number comes in. So this, you can see a very distinct result in the gamete production. So genetically, if we go with possibility 1, randomly they just happen to line up this way, we get these gametes. Randomly, possibility 2, we get a different genetic result in our gametes, in the daughter cells. And that is going to be solely from the allele differences between paternal and maternal chromosomes and how you inherit them. Okay? This hasn't even been talking, now we haven't even considered the degree to which recombinant chromosomes, or the result of crossing over, adds to that diversity as well. So let's take a look at that. So again, remember, you have one of your chromosomes in each pair from your mom, and one of the other chromosomes in each of your pair from your dad. But you don't want to hand off just the stuff from your mom or just the stuff from your dad. You want to hand off a little bit of both of your parents to your kids. Right? So we have crossing over. Tetrads form, synapses forms, we get crossing over events. Right? So that physical location where the DNA is being broken and reknitted to the new chromosome is called a chiasma. Right? And you can get in, when you take a more advanced molecular biology class, you'll talk about holiday junctions and all this kind of stuff. But we don't have to worry about that now. Literally, there are cool proteins and mechanisms to say, this is where I'm going to break the DNA, and I'm going to put it over in the exact same place where I broke it over here on this other chromosome. And what do we get? We get chromosomes that are mixed up now, literally mixing up. This is truly what sexual reproduction is all about, mixing up the genes right here on a cellular level. So in phase 1, they line up in pairs and then pull apart. And now look at the variety that we see in the daughter cells. All right? Considerable, because now on a chromosomal level we have variation, not just in the combinations of chromosomes, mom or dad, that you get into a particular gamete. Now the final idea here is the idea of random fertilization. So if just based on independent assortment I can have 8.4 million possible chromosome combinations, different in any one of the gametes that is produced, if I pair that up with an equal number of variability in the egg, so let's say sperm and egg, it's 70 trillion diploid combinations. Because any given sperm can mix with any given egg that are any given one of the 8.4 million combinations of chromosomes just based on independent assortment. So it's 70 trillion diploid combinations. That basically says that the likelihood that anyone ever in the history of people is going to have the same combinations as you is not possible. So just like everyone else, you are unique. Now, again, what's the point? The point is that we want to maintain genetic diversity. Genetic variation, diversity in a population, is absolutely essential for evolution to work and for evolution to work robustly. If you want a population to survive in a changing environment, that's what you need, and that's why sexual reproduction is so predominant among eukaryotes.